This is Dr. Fuad Zwain. I'm an associate professor at the Department of Pharmacology and Toxicology here at AUB. I'll be giving you today a lecture about lipoproteins, disorders, and metabolism, and this will be a prerequisite for the drug of dyslipidemia lecture, which I'll also be giving. If you have any questions at any time, please feel free to email me at fz15@aub.edu.lb. And also, if you're interested in research, please feel free to add me on my LinkedIn or my Twitter account to follow on our research progress, and I'll be more than happy also to get to know you and follow your achievement.
So let's start by the learning objectives. By the end of this lecture, you should be able to identify lipoprotein's main constituents, describe cholesterol metabolism and circulation, we're going to touch base on that, and recognize different lipoprotein structure and function, list the lipoproteins and recognize their metabolism, and differentiate between different lipoproteins. So first, we're going to start by understanding the main constituents of lipoproteins, the main lipid constituents of lipoprotein. There's three main constituents, which is cholesterol, which I'm sure you're very familiar with, and we have triglyceride, which is a molecule of glycerol and three fatty acids called triglyceride, and we have the phospholipids here, which is also a glycerol with two fatty acids, and you have a phospho group. The phospho group is very important in lipoprotein because it gives it the hydrophilic aspects, allowing it to circulate freely in blood. So with that, now later in other slides, we're going to go more into the detail about the structure of lipoproteins and different type of lipoproteins and how they are metabolized. So a little bit of background on the two constituents of lipids in the body, the major ones, cholesterol and triglyceride, basically. So 2/3 of body cholesterol is synthesized in the liver. So we synthesize cholesterol. We don't take all cholesterols from food. It's synthesized from single acetate unit, and this is a major target for dyslipidemia drugs, which is the major group of drugs use statins. I'm sure you heard of statins before. So statins basically inhibit cholesterol synthesis by inhibiting this reductase enzyme, and we're going to see that later. Now, 1/3 of body cholesterol is ingested. That's why one of the main preventive therapy or non-drug therapy to decrease cholesterol is by having a lifestyle modification, including a decrease in a diet rich in cholesterol. So don't worry, if you stop cholesterol, you're not gonna die because your body is able to produce 2/3 of cholesterol. And we know cholesterol is vital for normal organ homeostasis because it's mainly a component of the cell membrane and precursor of bile acid for digestion and steroids, hormones, and vitamin D as well. Now, triglyceride is the main constituent of body fat. So this is how the body stores the fat in form of triglyceride. It's basic biochemistry and is derived from glycerol, and three fatty acids form the triglyceride. It's a great source of energy. There's no denying that. Usually, a lipase will hydrolyze the triglyceride and release the free fatty acids from the glycerol. Glycerol goes into a certain pathway to form either pyruvate or glucose, which is also a source of energy, and the Fa is degraded through lipolysis to C2 unit by oxidation, beta oxidation in the mitochondria that process it into ATP through the Krebs cycle. Moving forward, it's important for us to understand how our body synthesizes cholesterol at the level of the liver. It's actually synthesized by acetate, a combination of these molecules right here, which are transformed in a rate-limiting step by HMG-CoA reductase that converts it to mevalonic acid, is a precursor of cholesterol synthesis. Mevalonic acid is then processed through 20 steps to give us cholesterol. And cholesterol, of course, is the main constituent of bile acid formation. Now, why this slide is very important, because in the context of dyslipidemia and hypercholesterolemia, there's a very important enzyme here that is the target of one of the most prescribed dyslipidemic drug, which is statin. Statin acts actually by inhibiting this HMG-CoA reductase, reducing, therefore, the endogenous synthesis of cholesterol and reducing the level of cholesterol in the circulation. It's also equally important for us to understand the fate of cholesterol, a mechanism known as enterohepatic circulation. This is very famous. I'm sure you studied it in physiology. So technically what happens, you have 2/3 of your cholesterol synthesized and released in with the bile acid, we call it biliary cholesterol, and you have the 1/3 which is synthesized by the, not synthesized, ingested through diet, the dietary cholesterol. But both of them, when they go into the intestine, in fact, 50% of cholesterol is reabsorbed and the other 50% is excreted. That being said, this is a very important also target for drugs that inhibit the reabsorption of cholesterol and hence reducing the level of cholesterol in the circulation. And this is something we're going to talk about more in details in the lecture of dyslipidemic drugs. This is another summary of enterohepatic circulation that we just talked about in the previous slide. So here you have cholesterol that's basically synthesized by the liver and released in the intestine by the bile acids. As we said, 50% of it is reabsorbed, and the remaining 50% is secreted in the fecal matter. For the bile, on the other hand, 97% of the secreted bile is reabsorbed and only 3% is released in the fecal matter. Why this is important? Because you have to know that your system can realize when we have a decrease or an increase in cholesterol absorption. So let's say for any case we have an increase in cholesterol absorption with the enterohepatic circulation, the system will say, well, I have enough cholesterol here, so I need to reduce the endogenous production. And this is very important when you talk about treatment, because if you reduce the, if you basically try to manage hypercholesteremia by reducing cholesterol reabsorption, your system itself is trying to compensate that decrease and will try to produce cholesterol more from the liver. That's why later on you will see that dual therapy is very important. You can inhibit the reabsorption of cholesterol at the level of intestine, but if you don't inhibit the synthesis of cholesterol by using statin that inhibits HMG-CoA reductase, the compensation system can basically reduce the efficiency of the therapy. So now moving to the interesting part of this lecture, which is lipoproteins. So first we're going to talk about lipoprotein structure and classification before moving and studying the metabolism of each lipoprotein. The most important thing here that you need to know, the outer and the inner structure of lipoprotein is very important. So as you can see, you have apoprotein, which is protein attached to the outer layer of the lipoprotein. They play a major role in giving the lipoprotein a functional identity. We're going to talk about that later. And you have, of course, phospholipids on the outside. This gives the hydrophilic nature of phospholipid, allowing it to circulate freely in a circulation, along with unesterified cholesterol. Moving down to the inside, the hydrophobic core. Yes, at the end of the day, lipoproteins are nothing but carriers of lipid. It's very important. And how do they carry that? They put everything that's fat inside, hydrophobic, because you cannot have triglyceride really floating freely in the circulation. It has to have a carrier, and lipoprotein is that carrier, the core of lipoprotein. Same for cholesterol ester. Cholesterol ester is nothing but a molecule of cholesterol that is esterified with fatty acid, making it hydrophobic, allowing it to go and sit inside the core of lipoprotein. Because cholesterol in itself can circulate in the circulation under the form of free cholesterol, but once you esterify it with a specific enzyme, we're gonna see that later, it goes inside the core and it becomes a part of the lipoprotein. So let's start with classification of lipoproteins. They are classified on the basis of their density. If you have high density lipoproteins and low density lipoproteins. The high density lipoproteins means that they have more protein to them, and the low density lipoproteins means they have more lipids into them. It makes perfect sense. When you mix oil and water, what happens? The oil floats on the surface because it's way less dense. So more lipids you have, the lower is the density, right? So the main classes of lipoproteins are chylomicrons and chylomicrons remnants. The VLDL, also known as very low density lipoprotein. Now you can know when we say very low density lipoprotein, it means you have way more lipids. Low density lipoproteins, which is a product of the LDL, and HDL, which is the high density lipoprotein, which means that it has more rich proteins than a lipid. And we have lipoprotein. In this lecture, we'll be focusing on a few lipoproteins, the most important ones. We're going to be talking about chylomicrons and the important role in transporting the dietary triglyceride, and some cholesterol. And VLDL, very low density lipoprotein, which is very important in transporting the endogenous triglyceride and some cholesterol. We're going to see how. We have the IDL and LDL, both byproducts of VLDL, and are very important in transporting cholesterol ester. And we have HDL, which is, everyone knows HDL is the good cholesterol. Why is that? Because it ensures reverse transport of free cholesterol from the circulation back to the liver, and also play a major role in exchanging apoproteins with the other lipoproteins in the circulation. And we're going to talk also about that in detail. So we're going to basically break down each lipoprotein, discuss its metabolism in this lecture. And this is just a more organized slide that summarizes lipoprotein structure and function. So again, the main function of lipoproteins, they work as carriers. Okay, carriers of lipid. They possess a water-soluble shell, as we said. It's formed by phospholipids, and it serves to carry the hydrophobic cholesterol and triglyceride embedded inside the core and transport them into the blood to the target tissues, such as adipose tissue, muscles, and liver. The composition, as we said, a monolayer phospholipid shell is hydrophilic, allowing the lipoprotein to circulate in the circulation. It usually has apolipoprotein on the surface and unesterified cholesterol. And inside the core, the hydrophobic core, you will have cholesterol esters, which are a molecule of cholesterol and fatty acid together, triglyceride, and this is hydrophobic. The apolipoproteins are nothing but proteins, and they're very important for the structure and stability of the lipoprotein, as well as the function of lipoprotein. They actually activate enzymes that metabolize lipoproteins, such as lipoprotein lipase. We have different classes of apo, depending on the type of lipoprotein we're talking about. We have apo A, B, C, and E. And an example of that would be ApoB100, for instance, that deliver lipoproteins to non-hepatic cells. We're going to talk about this in details in the following slides. Before we jump into the detail of each lipoprotein metabolism, it is important for us to define two pathways, the exogenous pathway and endogenous pathway. Both are lipoprotein metabolic pathways. What is the difference between these two? The exogenous pathway is mainly based on all the lipids that we eat, dietary lipids basically. The dietary lipids go through the intestine and then it's processed into something called the chylomicrons. The chylomicrons takes the fat, triglyceride, and cholesterol and form this big lipoprotein, a very high density lipoprotein. It goes into the circulation where it's hydrolyzed by lipoprotein lipase and target tissue, mainly muscle and adipose tissue. And then it's converted as, the triglyceride level shrinks and cholesterol level increase. We have a chylomicron remnants that goes back into the liver to be further processed. The endogenous pathway, on the other hand, is based on hepatic lipid only. Hepatic lipid is assembled into a lipoprotein called the VLDL, or very low density lipoprotein, because it's very much rich in lipid, very high in triglyceride, less in cholesterol. And then it goes into circulation the same fate as chylomicron, and it's processed into IDL. IDL has a higher cholesterol, low triglyceride, because triglyceride has been taken by the tissue. And IDL either goes back to the liver or it's converted to what's known as LDL, very famous. You know that, the low density lipoprotein, very rich in cholesterol, that goes to the tissue and deliver the cholesterol to the tissue. So let's start with our first lipoprotein, chylomicrons. Remember, we said chylomicrons is produced from dietary lipids in the intestines, right? So dietary lipids are converted into chylomicrons. Chylomicrons is mainly formed by the structural apolipoprotein known as ApoB48, but also it has other important apolipoproteins such as ApoA1 and ApoC2 and ApoA. Now, ApoA1 is very important because what chylomicron does, it gives ApoA1 to a nascent HDL during the HDL formation in exchange for two other important apolipoprotein, the ApoC2 and ApoE. So ApoC2 and ApoE are taken from HDL in exchange for the ApoA1 from the chylomicron. Now, so when chylomicrons circulate in the circulation, start being hydrolyzed by lipoprotein lipase and reduce all the triglyceride content, it shrinks into a chylomicron remnant, very rich in cholesterol, where it goes back to the liver through the ApoE receptor. It goes into the liver where it's going to be further processed. Just to see the picture as a whole, after the lipid is ingested, it's basically emulsified by the bile salts, where we have a lot of intestinal lipase that degrade the triglyceride into glycerol and fatty acids. And fatty acids, the breakdown molecules, are taken by the intestinal mucosa right here, and this is where the chylomicron is formed. The triglyceride and cholesterol incorporated into the chylomicron along with the adequate apolipoprotein, and then the chylomicron itself will go into the lymphatic circulation, bloodstream circulation, to be delivered to the tissue. At the level of the tissue, in the capillaries itself, lipoprotein lipase is expressed along the capillary by the endothelial cells. It's activated by the ApoC2 of the chylomicron. Remember, the ApoC2, which is acquired by HDL in exchange of ApoA1, and then in the tissue, the triglyceride is processed into glycerol and free fatty acid and taken up by these cells. So this is just a closer look on what happens at the level of the intestinal lumen and intestinal mucosa cells in terms of chylomicron formation. So as we said, we have the mixed micelle that's formed by emulsification of lipids that comes from diet, and they have the triglycerides and free cholesterol. In response to lipase, they're going to be degraded, the triglyceride mainly to monoglycerol and the monoglycerol right here and free fatty acid. They move through the unstirred lipid layers into the intestinal mucosa cells. And free cholesterol is polarized, can move easily, so it goes straight on its own also to the intestinal mucosa cells without being digested. And inside the intestinal mucosa cell, you're going to have the fatty acid and monocyclic glycerol. They're going to be combined again to form triglyceride, and the free fatty acid and the free cholesterol together, they're going to form the cholesterol ester. Both cholesterol ester and triglyceride are hydrophobic, and they go into the core of chylomicron formation. Of course, chylomicron with ApoB48, as we said, we have this full chylomicrons, mature chylomicrons that's ready to go into the bloodstream and the lymphatic system to be further processed. I'm not going to further dwell on this. This is just a slide, a cartoon for you to see everything that we talked about a little bit earlier. And in the next few slides, I'm going to summarize it in writing as well. So mainly the chylomicron moves into circulation, acquires the ApoC2, ApoE, and gives away the ApoA1 to HDL, and through ApoC2 it binds to LDL, to LPL, the lipoprotein lipase right here, and then free fatty acid is taken up from the chylomicron. The chylomicron will shrink into chylomicron remnant and uses the ApoE to bind to the ApoE receptor, where it's going to be further processed and recycled inside the liver. So the next 3 slides are just dictating what I already told you about chylomicron's metabolism, so feel free to read them so you can test your knowledge accordingly. Nothing new here, same story, chylomicrometabolism in a summary. I like the slide because it talks to focus a little bit more on how the ApoC, ApoE, and ApoA are exchanged. You know, as we said, the nascent chylomicron, they acquire ApoC and ApoE from HDL to form the mature chylomicron exchange of ApoA that goes to HDL to form a mature HDL. And then chylomicron, after being processed through lipoprotein lipase, it loses the ApoC, and we have the chylomicron remnant with B48 and ApoE is taken by the liver through the ApoE receptor. The chylomicron remnant is taken by the liver through the ApoE receptor. With that, we finished the chylomicrometabolism. I hope you really learned the details of it, and we're going to move to our next lipoprotein. So our next lipoprotein in this lecture will be VLDL, or very low density lipoprotein. You know now that when you say very low density lipoprotein, it means it's very rich in lipid. But this lipoprotein is different from chylomicrons in the terms of its source. As we said, chylomicrons come from dietary lipid and is formed in the intestine. The VLDL, on the other hand, is formed endogenously by hepatic lipids. I will here liver synthesize the VLDL. The VLDL also have an ApoB protein. It's called ApoB100, very important for the structural stability of the lipoprotein, and also it has a very important role later on after processing VLDL into IDL and LDL. We're going to see that later on. Nascent VLDL acquires ApoC2 and ApoE from HDL, similarly to chylomicrons, go into circulation and start where it's recognized by lipoprotein lipase through the ApoC2 that activates the lipoprotein lipase. And we have hydrolysis of the triglyceride inside into free fatty acid and cholesterol, and glycerol is taken by the tissue. And as that happens after lipoprotein lipase step, ApoC2 and ApoE are given back to HDL. We're going to be left with VLDL remnant, which is nothing but an IDL. We're going to see that goes straight back to the liver, and or an LDL, a low density lipoprotein, which mainly contains cholesterol only. Remember, after we go through the lipoprotein lipase step, triglyceride levels will decrease, and we're going to be left with the bulk of cholesterol is there inside these lipoproteins. So just some additional information and a little bit of things that we said, a little bit of new stuff that we need to know. VLDL is very rich in triglyceride. It has some cholesterol. The major site of formation is in the liver, mainly it's endogenous pathway, hepatic lipids. The secretion of the LDL is related to dietary and hormonal status, and it is enhanced with fat state. Makes sense. The more lipids you have in your system, the more lipids going to arrive to the liver, the more VLDL the liver is going to form. So when you have also a high diet in carbohydrate, you know carbohydrate increase the rate of lipogenesis, formation of lipid, and high levels of free fatty acid in the circulation are taken back to the liver, where also they can be combined into triglyceride and embedded into a VLDL formation. So the main role of VLDL is the transport triglyceride from the liver to the extrahepatic tissue and muscle, adipose tissue, and cells in need. And then, as we said, triglyceride is going to be hydrolyzed by LPL through the ApoC2 activation site of the lipoprotein, and it will be converted into IDL and LDL. A very important concept that we're going to talk about later in details, the CETP in HDL. This is an enzyme that allows the exchange of triglyceride from VLDL with cholesterol is there from HDL. This is another way how HDL can transport cholesterol back to the liver, and we're going to see that later on in the next slide, so don't worry about this. Last sentence, we're going to explore it further in details later on. So again, here it's we just repeating some stuff. It's normal. The mature VLDL, as I said earlier, they contain the ApoB100 plus ApoC2 and ApoE from HDL, as we said. And ApoC2 is required for the activation of lipoprotein lipase. Most important part of this slide is that VLDL, when it's hydrolyzed by LPL, is converted to a VLDL remnant known as IDL. And once ApoC is given back to HDL, it becomes smaller in size and becomes more and more dense, because it's very rich in cholesterol and very, very less rich in triglyceride. So when you lose a lot of triglyceride, losing a lot of lipids, we know lipid make things less dense, the remnant will automatically becomes more dense. The end product, the IDL, when it returns the ApoE to HDL, it becomes an LDL. So at this point, the IDL can either go straight to the liver, bind to the ApoE receptor where it's internalized and processed, or it could give the ApoE to HDL and become something called the LDL. Low density lipoprotein is a very important biomarker in terms of dyslipidemia when we do blood tests. Its most core lipid is cholesterol ester, and it only have one lipoprotein, which is ApoB100 on its surface. It's very important to be recognized by the LDL receptor. We're going to talk in details about that when we reach the slide of LDL metabolism. A very important concept that we must touch base on is the exchange that happens between HDL and other lipoproteins in the circulation. And as we're going to see in the drug for dyslipidemia lecture, we're going to talk about that and highlight on the importance of this mechanism in restoring the HDL activity in case of dyslipidemia. But this is not what we're going to talk about today. Today we're going to just, in this lecture particularly, we're going to just highlight what really happens. There's an enzyme called the CETP enzyme that allows actually HDL to exchange cholesterol ester, as you can see here, cholesterol ester given to VLDL in exchange for triglyceride. And this is a way where HDL is able to get rid of cholesterol either by taking it all the way to the liver, as usually it does, or it could exchange it with VLDL, and VLDL it goes all the way to LDL and it's coming back to the liver as well. So this is two ways how HDL can bring cholesterol back from the circulation all the way to the liver. So this is a very important mechanism that you guys need to know. Just the VLDL metabolism. This is the last slide, VLDL metabolism. Just a summary that mainly highlight all the points that we talked about, from the formation of VLDL and the importance of apoproteins B100 on VLDL and the C and E that are acquired from HDL, to the circulation of VLDL and how it's hydrolyzed by lipoprotein lipase through the recognition activation site of ApoC2, and then after it's hydrolyzed, it becomes a VLDL remnant known as IDL that has ApoB100 and ApoE, because it gets rid of ApoC during this process, given back to HDL, and then IDL is converted to either converted to LDL by getting rid of ApoE, or it's taken directly by the liver through the ApoE receptor to be further processed. And then what happens to LDL? We're going to see that in the next slides. Move to our third lipoprotein, which is LDL. Since we talked about chylomicrons and VLDL, I think now you have a general idea about what LDL might be. So LDL is nothing but a byproduct of VLDL. So technically, as we said, once VLDL is formed, it goes into circulation, undergo lipolysis by lipoprotein lipase, and we have the VLDL remnant formation, also known as IDL. And IDL will give away ApoC to HDL, and through its ApoE, it can go straight to the liver, where it's get internalized and metabolized, or it could give away the ApoE and it's converted to the LDL, the low density lipoprotein, which goes into the circulation to be taken by tissues that are in need of cholesterol through the LDL receptor, or it could be cleared by the liver through the ApoB100 receptor. The only Apo that LDL has is the ApoB100. This is just the written information of what we just said. It's the major cholesterol carrier in the blood. It has only one apoprotein, ApoB100, formed from VLDL remnant IDL by getting rid of ApoE and giving it back to HDL. It carries cholesterol to special tissue and recognized on the cell surface by receptor to ApoB100. Once it binds to this receptor, it undergoes endocytosis, and we're going to see that. Before we talk about the fate of LDL, it's important to talk about the LDL receptor, because the LDL receptor itself is the main target for the dyslipidemia, as we're going to see in this lipidemia lecture. So the LDL receptor is also known as ApoB100 and ApoE receptor. Why? Because it recognizes both ApoE and ApoB100. However, the affinity for ApoE is much higher than the affinity of ApoB100. So if you have more ApoE in the circulation, the receptor, it binds to a larger extent to ApoE rather than ApoB. With that being said, the LDL itself that has ApoB100 has low affinity to the LDL receptor, especially in the liver. The complexes of LDL are taken into the cell by endocytosis. Once they bind to the LDL receptor, LDL and LDL receptor got internalized. We're going to see that and what happens. And the important role of LDL is that it highly correlates with bad cholesterol. So whenever you have high LDL level in a blood test, you would know that, well, if you have high LDL level, it means that the cholesterol level is high, and this correlates a lot with high risk of cardiovascular diseases. Another important point is the saturated fat. If you have high saturated fat and high saturated diet, it blocks the LDL receptor, which basically decrease the level of LDL uptake, increasing therefore LDL level in the circulation. And when you have a block of LDL receptor, the cells themselves will say, well, I have low cholesterol, so I need to tell the liver to produce more cholesterol. And this by itself will also increase as a negative feedback the level of cholesterol in the system, making things worse. Of note, familial hypercholesteremia is due to a mutation in LDL receptor, and as we said, subsequently, if you have a decrease in LDL receptor activity, this will stimulate the system to produce more cholesterol, and hence you're going to have a hypercholesteremia. And there is treatment for that, and we're going to see this in the drug for dyslipidemia lecture. ApoB100 recognizes the LDL receptor on the tissue. Once it binds, it gets internalized. Right here, the whole thing gets internalized, and it either go to the lysosome where everything is degraded. So you take the LDL and you break it down into everything that contains the phospholipid, the apoproteins, the cholesterol, everything, and the cholesterol and everything, the amino acids, fatty acids, and the cholesterol is used for the cellular homeostasis. And the receptor itself, it gets recycled and go all the way back to the surface. This mechanism of recycling the LDL receptor is also a very important target for drugs. So there are drugs on the market, particularly PCSK9 inhibitors, that decrease the degradation of LDL receptor and increase the recycling of the receptor, making receptors more readily available to collect the LDL from the circulation, and hence decreasing the LDL level in the plasma. A lot of questions about what is actually the fate of LDL cholesterol, and that's why I decided to put this slide to touch base on that. So non-hepatic cells, they obtain mainly cholesterol from the LDL that's circulating in the circulation. So LDL is internalized, and we take the cholesterol esters that is in it, and inside the lysosome, it's converted to free cholesterol. And this free cholesterol is either used anesterified for the biosynthesis of the cell membrane itself, so to maintain the integrity of the cell. You know, cholesterol inside the cell membrane is very important for the fluidity of the membrane. Reesterified for storage inside the cell by ACAT. It's an enzyme. We're going to see ACAT and LCAT are very important in the esterification of cholesterol and the storage. And also, I know that you all know that it's very important in the synthesis of steroids in specific tissues. So here we're going to try to understand what happens when the level of LDL in the blood, they go ORI, which means they go into abnormal levels, high levels, and this is a sign of dyslipidemia. And what happens in that case? What happened when the LDL level in the blood really increase, and why? First, we talked that the LDL receptor is basically has lower affinity for ApoB100 than ApoE. That being said, IDL that have ApoB100 ApoE can be easily taken up by the liver through the ApoE high affinity receptor, and we're left with LDL that have only ApoB100 that have a lower affinity. So in the presence of high IDL level, for instance, the LDL receptors can be saturated, and leaving us with LDL that has low affinity and could be, remain in the circulation and increased in level. There is a lot of course, reasons why LDL level is increased, not only because you have high IDL level. You have to ask questions, why do we have high IDL level to begin with? Because we have very high VLDL formation, so on and so forth. So the high LDL level per se, if it stays in the blood for a long time, it gets oxidized. And when it gets oxidized, the ApoB100 and the lipid inside the LDL undergo peroxidation, and they become basically a very good target for what we call the LDL scavenger receptors. These scavenger receptors exist on blood vessels, most prominently on macrophages in blood vessels. And once they take this oxidized LDL, they keep taking it and trying to digest it, and they transform into what's known as macrophages foam cells. And this is the basis of atherosclerotic plaque formation. So that's why high LDL level for a certain period of time, for a long period of time in the blood, increase the risk of LDL being oxidized, increase the risk of it being taken up by macrophages, and this will lead to a foam cell formation, a trigger inflammatory reaction that eventually will lead to an atherosclerotic plaque formation and increased risk of cardiovascular disease. This is just a summary. We showed the slide before and nothing has changed here. It's just a summary of how VLDL is formed, how it transformed to VLDL, how it gives away the ApoA in exchange for ApoC and ApoE from HDL, and how it's taken up by lipoprotein lipase to be then hydrolyzed into the VLDL remnant, the IDL that has the ApoE and ApoB100s. Through the ApoE high affinity, it can go into the liver that has the LDL receptor with higher affinity to ApoE, or it could give away the ApoE back to HDL, and we end up with the LDL with ApoB100 that has lower affinity to the LDL receptor, that's either taken up by the liver or it goes into extrahepatic tissue, but also bind to LDL receptor and it gets internalized and processed this way. Now we reach the final lipoprotein of today's lecture. It's the good one, actually, the high density lipoprotein. Now, why is it high density lipoprotein? Because it's mostly rich in apolipoprotein, and it doesn't really have high level of lipid compared to the other lipoproteins that we talked about. So there is a process of maturation of HDL. We're going to talk about it. The formation of HDL is known as nascent HDL, and then it goes into different stages as it acquires cholesterol through esterification. There's a very important enzyme that we're going to talk about now, known as LCAT. That's important in the esterification of unesterified cholesterol to cholesterol esters to be embedded inside the HDL, so it can be transported back to the liver or used to be exchanged with a triglyceride from other lipoprotein. We're going to see all of that in a bit. Here, let's touch base briefly on HDL functions, different HDL functions. We did talk about that in different slides, but let's try to remind ourselves of what HDL really does. First, you have the transfers of protein to other lipoprotein. And this is a concept that we talked about. We said that HDL transfer ApoC and ApoE to VLDL and the chylomicron, and it picks up ApoA1 from chylomicron itself. And apolipoprotein are very important for structure, for stability, and for the function of lipoprotein. It picks up lipid from other lipoprotein, the concept of CETP that we talked about. It gives away cholesterol ester to VLDL, for example, and takes triglyceride. This is another way for it to bring cholesterol back to the liver. It also picks up free cholesterol from cell membrane. A lot of cells are either dying or in renewal, phase, they release cholesterol. The circulation of free cholesterol is esterified and embedded into the core of HDL and brought back to the liver. And this is the ALCAT, the enzyme. We're going to talk a little bit about it later, just brief comparison between ALCAT and ACAT and the concept of cholesterol esterification. The HDL can transfer cholesterol ester into the liver into the concept known as reverse cholesterol transport. What is reverse cholesterol transport? It's simply the mechanism by which HDL picks up cholesterol and brings it back to the liver. We call it reverse cholesterol transport. And now we know that it exists in two major ways, either directly, so HDL itself going back to the liver, or indirectly through other lipoprotein by giving the cholesterol ester in exchange for triglyceride to other lipoprotein. For example, it gives it to VLDL, and VLDL is converted to IDL and LDL, and cholesterol is brought back to the liver, or through LDL taken to other tissues. The high blood level of HDL, excuse me, is known as the good cholesterol. And you know, when someone has high HDL, it means that, well, he has the good cholesterol, simply because it reduces the level of cholesterol in the circulation. There's a lot of therapies. We're going to talk about some of the therapies in the dyslipidemia lecture that increase the level of HDL, basically. And high HDL level correlates well with low incidence of atherosclerosis. Slide simply summarize the mechanism of reverse cholesterol transport, by which HDL right here can either take the cholesterol, the esterified cholesterol that's in the core right here, and takes it all the way to the liver to be processed, or through the CETP enzyme. We talked about that. It exchanged the esterified cholesterol with VLDL in exchange of triglyceride. And also it can, of course, acquire the unesterified cholesterol through its esterification by ALCAT. The free cholesterol is esterified and it's embedded in the HDL. The point here is to have two ways, either directly by taking the cholesterol to the liver, or indirectly by exchanging it with other lipoproteins such as VLDL. So nothing new in this slide. We are just repeating things and strengthening our knowledge at this point. Nascent HDL itself consists of discoid phospholipid bilayers. It's containing ApoE, ApoC, and free cholesterol. Now, once they enter the circulation, they acquire ApoA from the chylomicron. We did say that already, and this is the ApoA is a major structural HDL apoprotein, and they give away ApoC and ApoE to CM and VLDL, which are very important for their own metabolism as well. ALCAT is a very important enzyme in HDL, activated by ApoA1 and binds to these discoidal particles and the surface phospholipids and free cholesterol. They convert them to cholesterol ester and lysolecithin. And the esterified cholesterol is embedded inside the core, the hydrophobic core of HDL, to be transported back to the liver. So the nonpolar CE move into the hydrophobic anterior tubular layer, and a spherical HDL is formed. It's called the mature HDL that acquired as much as it can of esterified cholesterol, and it's taken back to the liver to dump that cholesterol in a mechanism known as reverse cholesterol transport. This is just a graphical summary of what we just said. So you can use it and to visualize what we talked about, so you can better understand the concept of HDL metabolism. So this is just a summary of all the lipoproteins that we talked about. You can use this as a reference slide. We have the chylomicron that's synthesized in the small intestines from the lipids that we digest, that we eat, the dietary lipids. It has different apolipoproteins on it. We have ApoA1, which is transferred to HDL, important for HDL metabolism. The ApoB48, remember, it's important for the chylomicron structure and stability. The ApoC2 for lipoprotein lipase activation to hydrolyze triglyceride. And ApoE is actually the remnant receptor binding, so the chylomicron remnant to be taken back by the liver to be metabolized there. The VLDL, on the other hand, it's synthesized in the liver from endogenous lipids. So whatever lipids that are synthesized in the liver, the triglyceride and so on, they are put together to form the very low density lipoprotein. And the VLDL has ApoB100 on it. It's important receptor binding. Remember that the ApoB100 has lower affinity than ApoE for the LDL receptor. The ApoC2, same as chylomicron, for lipoprotein lipase activation to hydrolyze triglyceride in the peripheral tissue. And ApoE is the remnant receptor binding. Remember, the VLDL remnant receptor is going to be IDL. It has only ApoB100 and ApoE on it, and it's important to take IDL back to the liver. Now, if IDL give away the ApoE, it becomes an LDL, the low density lipoprotein. So that's why it's synthesized from IDL. LDL is very important in cholesterol transport. Remember, the LDL reflect the bad cholesterol if it's high. It has only one receptor, the ApoB100 on it. And remember, the ApoB100 has less affinity to ApoE for LDL receptor. And then we move to HDL, the good cholesterol, the high density lipoprotein. It's synthesized in small intestine and liver, and it's important in reverse cholesterol transport. Why? Because it brings cholesterol from the circulation back to the liver. It has ApoC2 and ApoE on it, which is usually transferred to the LDL and CM, which is important for their metabolism. And it has ApoA1, which is taken from the chylomicron, and ApoA1 is an important cofactor for LCAT. Remember, LCAT is an enzyme that esterifies cholesterol into cholesterol ester, so it can be stored in the core of HDL to be brought back to the liver. Here we're going to touch base on a few enzymes that are important in metabolism of lipoprotein in the next two slides. So let's start with lipoprotein lipase. We said this is an enzyme. It's very important to hydrolyze triglyceride from the chylomicron and VLDL. It's attached to the luminal surface of endothelial cells in the capillary, so it can bind directly to the moving VLDL and chylomicron in circulation. It's highly expressed in the heart, muscle, and adipose tissue, and then it's taken up to the endothelial cell on the surface. It hydrolyzes triglyceride into free fatty acid and glycerol, as we said, from VLDL and chylomicron. The hepatic lipase, it's similar to LPL, but it exists on the liver cells. And it also hydrolyzes triglyceride into free fatty acid and glycerol from everything that's coming back to the liver. However, unlike unlike LPL, the hepatic lipase does not react readily with chylomicron and VLDL, but is concerned with triglyceride hydrolysis from the remnant of VLDL, such as IDL, which is IDL, and the chylomicron remnant and HDL metabolism. Three additional enzymes are important in the metabolism of lipoproteins. We have ALCAT, which is important in the formation of cholesterol esters for storage in HDL. Remember, this is an enzyme that uses ApoE1 as a cofactor to take the free cholesterol, esterify it, and put it inside HDL in the circulation. The ACAT is similar to ALCAT, the same exact thing. However, it's more involved in the formation of cholesterol ester for storage in the cells, not the HDL that is in the circulation. And finally, we have CETP. CETP, as we said during this lecture, it's very important in reverse cholesterol transport, and its role is to facilitate the exchange of cholesterol ester and triglyceride between lipoprotein. So a quick summary, which lipoproteins carries mainly exogenous triglyceride to peripheral tissues? It's chylomicrons, right? Which lipoproteins carries mainly endogenous triglyceride to peripheral tissues? So the endogenous triglyceride is triglyceride formed by wrapped up in the liver, basically, and delivered to the tissue. So here we're talking about the very first one that's released from the liver, and it's called VLDL, right? Which lipoproteins carries mainly cholesterol to peripheral tissues? So here we're talking about the lipoprotein that is metabolized from VLDL. You know, VLDL goes through, deliver the triglyceride to the tissues, is converted to IDL, and IDL is unstable. 50% of it goes to the liver, and the other 50% is metabolized by hepatic lipase into the LDL, the low density lipoprotein that binds to the tissue, the LDL receptor on the tissues, internalize and release cholesterol inside the tissue. So here we're talking about LDL. Which lipoproteins carries mainly free cholesterol back to the liver? You know, when we have excess of cholesterol that the cells don't need, they release it into the circulation, and it gets picked up by which lipoprotein? The good cholesterol, HDL, that takes it back to the liver.